Hello friends, welcome to classes by Lab Doctor. This is Dr. Divya and in today's class let us study about the semen morphology. When I say semen morphology, इसका मतलब क्या है? It is the shape of the sperm. It is the appearance of the sperm which can be abnormal, and because of this abnormality it can affect its capacity to fertilize a woman's egg. So sperms are classified as abnormal shapes based on the defects in the head or defects in the tail, defects in the midpiece, etc. But the WHO says that the lower limit of the normal for the sperm morphology is 4%. That means अगर हम एक 100 sperms को काउंट करते हैं, उसमें से अगर 4% भी नॉर्मल रहे, उनका हेड नॉर्मल है, टेल नॉर्मल है, मिड पीस नॉर्मल है, that is still better. So the lower limit is around 4%. And when the morphology is very bad, couples still have the choice of using IVF, IUI, ICSI to improve the chances of becoming pregnant. So the sperm morphology, we are going to study the shape of the sperm, the appearance of the sperm, which includes the head size, which depends upon the head DNA content, the midpiece, and the structure of the tail. And it is very important that we assess this because it is the head of the sperm that penetrates into the outer surface of the woman's egg and fertilizes it. सो ये जो नॉर्मल मॉर्फोलॉजी कैसा होगा उसके बारे में पहले हम डिस्कस करेंगे. सो एक जो नॉर्मल स्पर्म रहता है उसमें हेड रहता है and this is the head which consists of nucleus, यहाँ पे रहेगा, and it which is having a condensed chromatin, and nucleus में sometimes there could be nuclear vacuoles, and this is the acrosome. ये जो cap है आप देख रहे हो, that is the acrosome. So the head consists of the nucleus as well as उसके ऊपर जो cap है. जो ये acrosome है, anterior 2/3 of the head will be this acrosome. That means अगर आप ये head को देखोगे तो उसमें से around 40 to 70% of the head will be composed of this cap or the acrosome. And this acrosome, it is important because ये some enzymes को secrete करेगा which will dissolve the layers of the ovum and it will help in the penetration. Then you have the middle piece which contains the mitochondria which provides energy for the movement, and the tail is the one which will help in the motility. So this is the head, this is the nucleus, this is the acrosome. So the head consists of nucleus and the acrosome. This is the intermediate piece, or this is the tail or the flagella. So you will sometimes hear the principal piece, terminal piece, etc. So remember, the sperm will have head as well as the tail. So this is the head and this is the tail. This part, the tail part, is divided into mid piece, principal piece and the terminal piece. So this is the middle piece, this is the principal piece and this is the terminal piece. So basically उसमें head रहता है and tail रहता है, and tail में हम ये तीन category mid piece, principal piece and terminal piece बोल के we will divide it. So this is the acrosomal cap, this is the acrosome, this is the nucleus, ये nucleus का membrane है. Here you can see the mitochondria. This is called as the dense fibers. This is the exoneme. This is the annulus, the ring-like structure, and these are the axial filaments. So what are the requirements? अगर ये normal sperm है बोलने के लिए हमको क्या requirement चाहिए? So first thing is that the sperm should have a smooth rimmed, जो head रहेगा वो smooth होना चाहिए, and it should be oval in shape. And head का जो size है, it is usually between 2.5 to 3.5 micrometer wide and 5 to 6 micrometer इसका length रहेगा. And this acrosome, what is there? Acrosome is nothing but it is a cap-like structure which is a membrane wherein the enzymes are capable of penetrating. This will cover the 40 to 70% of the sperm head. ये normal है. This head should not have any large vacuoles या उधर कोई भी fluid filled structures नहीं होने चाहिए. And अगर रहते भी है, then there should be not more than two such vacuoles, and they should be taking up less than 20% of the head. So this is about the normal head of the sperm. What about the mid-piece of the sperm? This is a segment between the head and tail. It should be the same length as the head. It is little slimmer. This is much more oval and wider. This is almost the length is same, but it is slimmer. It would also have a tail which is uncoiled, and it is around 45 micrometer long. And this has to be free from all the head, tail and mid-piece defects. So see, this is how the sperm develops, and whenever there is any problem in this kind of a development, there could be a abnormal morphology. So here you can see this is the nucleus of the spermatid. Then you will see that these are the centrioles which are present here. This later develops a flagella like this, and then the further development starts. The microtubules start growing, the mitochondria is formed here, and here you can see that it is now becoming a sperm. It is forming a nucleus, it is forming an acrosome. This is the mitochondria in the mid-piece, and this is the excess cytoplasm here, and this is the tail which is still developing. So here you can see the nucleus, it is becoming smaller, the acrosome is becoming bigger, and this is again the excess cytoplasm which is there. This is the mid-piece, and once it matures, you will have the head, you will have the mid-piece here. This is the tail. So there will be no excess cytoplasm. Majority would be the acrosome, the smaller part would be the nucleus. There will be no excess cytoplasm, and this would be around 45 micrometer tail. So this is how the spermatozoa is developed. So this morphology becomes important especially in cases of your artificial reproductive techniques. It is a very complex parameter. It's a difficult parameter. Abnormal morphology can happen because of abnormal spermatogenesis or sperm maturation. So in the previous slides, what you have seen here, the sperm maturation, if there is any abnormality in this process, you might find that the morphology of the sperm is also abnormal. There could be an impact on chromatin condensation, acrosome reactions, sperm motility, etc. So for morphological analysis, we usually prepare semen smears, and we assess the morphology. And WHO has given the lower reference value of 4%. That means at least 4% of the sperm should be normal, and if there are less than 4% of sperms which are normal, that condition is called as TERATOZOOSPERMIA. So TERATOZOOSPERMIA is a condition in which the man will be having more than 96% of his sperms are abnormal. It could be a defect in the head or in the mid-piece or in the tail, and less than 4% of his cells are normal. So this condition is termed as TERATOZOOSPERMIA. So the normal range as per WHO, I have told you that it should be at least 4%. So the chances of fertility will be high if the normal sperms are more than 14%, and the chances of fertility slightly reduces when the normal sperms are around 4 to 14%, and the chances of fertility or the chance to have a baby greatly diminishes if the normal sperms are less than 4%. It is just not the morphology. We have to take into consideration various other factors like sperm count है, sperm concentration है, कितना sperms alive है, कितना sperms move हो रहा है, the volume, the pH, the liquefaction, the viscosity, all these things will matter when we say the capacity of the sperm to fertilize. It's just not the morphology alone. And what could be the causes of abnormal sperm morphology? See, it could be genetics, it could be varicocele, it could be excessive alcohol abuse, it could be smoking, it could be hormonal imbalances, diabetes, stress, infection, lifestyle factors. All this could cause abnormality in sperm morphology. But at least 4% of the sperm should be normal to increase the capacity of fertilization and the chances of getting pregnant. So in the next classes we will see how do we assess the sperm morphology. So thank you so much for watching the video. Please like, share and subscribe the channel. Thank you.